All right, good afternoon, ladies and gentlemen. Welcome to our 14th installment of the Adventures in Field Research Seminar Series. We are super excited to have Dr. Stephanie Koester from Randolph-Macon College. She's going to give us a little presentation today called Close Encounters, Field Adventures of a Wildlife Biologist. And just as a reminder, we will have a Q&A session following the presentation. So if you have any questions, be sure to write them down. And if you cannot remember them or you don't have anything to write down, just type them in the chat and we'll get to them at the end. But with that, Dr. Koester, I'm going to pass it off to you.
All right, thank you. So I'm going to share my screen now. And this talk is going to be part kind of storytelling and part adventure sharing. It's going to be part advice giving, and it is going to be part science. So I hope you guys enjoy that. I'm going to start off by giving you a little bit of background about where I am from and what I do. So again, Dr. Stephanie Koester. I grew up in Maryland, just outside of Washington, D.C., in a pretty suburban situation. We're not talking rural. I didn't have tons of kind of exposure to wildlife in just by walking outside my door. There was lots of suburban homes and flowers and whatnot. But I did read, I read a lot. And one of the things I often read was National Geographic. And I always kind of wished that one day I could end up in the pages of National Geographic. And so I think that's kind of where some of that passion initially came from for thinking about animals and science and adventure, right? That all came together. And I was a Girl Scout all throughout these years, much later than it was still cool to be a Girl Scout. And the reason that I did it was I wasn't so interested in the service or the kind of, helping out or elderly or whatnot. Well, I did that and that was fun. What I really enjoyed about Girl Scouts was the outdoor adventures. And so here's actually a picture of me on my first rock climbing adventure through the Girl Scouts. We went up about 10 feet and then that was it. But I remember my heart was pounding the whole time and I was like, this is what I want to do. So I stayed in Girl Scouts until I was a senior in high school and did lots of kind of cool outdoor activities in that position. So with that, that's a little bit of a background on me. And I'm going to kind of switch to talking about some stories related to all my adventures that I've gotten into as a field biologist. So the first kind of advice, this is part advice. I don't know if I told you that, but it's also part advice. The first advice that I would recommend is if you're being a field biologist, choose your organism wisely. So you have to think about where these organisms are going to be, what time of day they're going to be, what the weather is going to be like when you're studying them. And that can all give you a really interesting perspective on what you want to do. So here's kind of pictures that I'm going to talk about in more detail today, and we're going to go through them and talk about the pros and cons of different organisms. So this is a picture of a class that I took just last year, pre-COVID, to Costa Rica, and we studied bats for a night, and so that was fun. But my first experience with fieldwork was as an undergraduate at Trinity University, and I helped out an upperclassman who was doing a research project with bats. And what we would do is we would go and drive out to the field site around kind of dusk. So it was just starting to get dark, but we still had some light. And then we would set up these nets, kind of like volleyball nets, but they're made of gossamer fine kind of strings. And we would set it up over usually kind of some kind of water system stream. And then we would sit there and wait until the bat came out. And so here we get into the pros and cons of bats. One of the pros of working with bats is that they are small and furry and that you can hold them. If you have the chance, it's very possible to be able to hold a bat when you capture it. You have to take them out of these nets. So that was pretty cool. I really enjoyed being able to hold organisms. That's top priority when you're thinking about being a wildlife biologist. You want to be able to do something where you can hold. So keep that in mind. And here's a picture of, this is again from the Costa Rica trip, but here's a picture of kind of a larger bat, and here's a picture of a smaller bat. So they come in all different sizes, also very fun when you're thinking about organisms to choose to research. And they also make little squeaking noises when you catch them. So that also, you know, that's a pro as well. Now, the cons of studying bats are that you really do have to be a night owl. You have to, so when we would set up these nets, we would set them up, and as I said, it was around dark, and then we'd have to stay out there till maybe about 1 a.m. And then we would pull all of the nets down and drive home, so we didn't get back until like 2 a.m. And that, my body clock could not handle that. It totally wrecked me forever. I'm not a night owl. I'm not sure I'm a morning person either. I guess I'm a day person. So I realized pretty quickly that even though they were fuzzy and made cute noises and you could hold them, I didn't think that these were going to be the organisms that I was going to study long term. So then I switched over. I did some traveling after undergrad, and I guess we'll talk a little bit about that later on. But then I came back to the United States and did a master's project at the University of New Hampshire, where I was studying black bears. And one of the pros about studying black bears is that they are large mammals that are very charismatic. So when you tell people that you study black bears, everyone is going to be like, oh, that's so cool, that's so fun. And that is always nice to get a good reaction when you're studying things. Also, another pro to studying black bears is that they evolved in a time and a place where they were not the top predator. So therefore, that means that when you scare them or when you interrupt them doing feeding or whatnot, they are going to run away from you, often crawling to the top of a tree. And that is a great, also as a field biologist, when your organisms are scared of you, it's a little bit better than when they are threatened by you. If it was going to be a grizzly bear, they might come after you. So again, that's a pro for black bears. They're going to be looking at you from a distance and not really attacking you. So another pro about black bears is that studying black bears is that very occasionally, if you're super lucky, oh, that was funny. That's not what it looks like. You get to, anyway, there's a picture under there of holding a bear. I don't know why it's not there, but anyway, there is. You'll just have to trust me on that. There's a picture under there of holding a bear, so that you occasionally get the opportunity to hold a black bear. And that was the picture on the slide that I was introduced with. So it's me holding a cuddly black bear and kind of keeping it warm. And I want to tell you a little bit about the story behind that. So this was a scenario where I had been studying black bears, and I had been working closely with a bear rehabilitator who was interested in some bears that he had raised by hand and let out into the wild to roam around the forest. And every winter, he goes out with some people from the Fish and Game Department, and they track these bears to their denning grounds. And then they interrupt their sleep in order to weigh them and measure them and get a sense of how much fat they have on their body and how healthy they are and how well the food resources were that year. And so here's a picture of us going out there and doing that. We had a radio tracker, and we were kind of tracking the signal for this bear who's wearing a radio collar. And then we found the bear underneath the rock, this like kind of rock ledge, and the Fish and Game person actually ended up tranquilizing it using a blow dart. So they blow dart and it tranquilizes the bear. And then we pull the mother bear out, and they're doing some kind of weighing and measuring and that kind of thing. And so during that time, we also have to measure all the baby bears that are in there. So here's a picture of the baby bear getting weighed in a backpack. And when the baby bear is done, what do you have to do? But you have to hold it inside your coat because it needs to stay warm, because it's used to being around its mother. It starts shivering. So when you're holding the baby bears, you have to put them inside your coat and keep them warm. Again, this is a pro about studying black bears. Pretty cute. Now, the one thing that is a con that you don't know from pictures when you see cute little baby fuzzy black bears is that they smell so badly. They smell like wet dog times 100. It is so stinky, but you just can't help it because they're in there and you're cuddling them. So something to think about if you don't like the smell of wet dog. And then finally, what we did was we put the mother back and the baby, and we, crept away and left them alone. So that was one of the kind of my close encounters with black bears. Now, oh, here's the picture. There's a picture that I was supposed to be showing you. But now I'm going to tell you a little bit about the science with this research project. So in this research project that I was doing, I actually wasn't holding black bears the whole time, even though I wished I was. As you can see, it's pretty intensive energetically to go and capture and measure and weigh these kind of black bears. And so what I was interested in for some of my research was, we were looking at an area of New Hampshire, these two different places, Pittsburgh and Milan. And yes, that is how you pronounce it in that area of the world. And they were interested in, the Fish and Game Department was interested in the population size of black bears in these two areas, because they had typically had a lot of bear sightings in Milan, which was a little bit more developed, but they didn't have that many sightings in Pittsburgh. But they were thinking about opening up the Pittsburgh region, which was mostly forested, kind of managed forest. They were thinking about opening that up to more hunting. And so they wanted to get a baseline estimate of how many bears were there before the hunting regulations changed for that area. And as you saw, the kind of capturing of the bears can be quite traumatic. So for this study, we did not do a kind of traditional capturing program where you would have these bear traps like this, but instead we used a mark recapture analysis with genetic techniques where we didn't actually ever have to capture the bear. So here is how it worked. What we did was we set up a barbed wire fence around the four edges of a tree. And then we hung a string in the treetop, and we had a little piece of fabric square doused in a scented lure. So it smelled like cherry flavor, very strong cherry flavor. And we hung that up in the trees, and that smell would carry off over the wind, and the bear would pick up that smell from a far distance and travel to figure out where that smell was coming from. And then when it got to the barbed wire fence, it would either climb over or under the barbed wire to eat the corn that we had left in there. And so that way, when it went over the barbed wire or under the barbed wire, we would snag hare. And so every day we would go out and pull up all the hares from the traps and mark them and catalog them, and just amass a tremendous sample. I think we had something like 1,700 samples of hair. And then I took it back to the lab where I extracted the DNA from the follicles and analyzed that DNA to figure out detect different individuals, and then try to figure out using different algorithms how many individuals were in that population. So here's an interesting sneaky story about these bears. Some of these bears were so smart, and I told you they were remember, they're not very aggressive. So what did we found out when we set up some camera traps like this one? We set up some camera traps near these sites, and we found out that we would go and visit a bear site and go and collect the hares every week around the same time. And then we would go and replenish the corn to make sure that that corn was available for bears coming in. And when we looked at our watches and took note of when we replenished the site, and then we went back and checked out these cameras, we found out that this, especially at one site, there was this one site where this bear must have been sitting there waiting for us week after week. He knew exactly what time we were arriving. We never saw him in the distance, hiding out, and he would come immediately after we left, and he would eat all the corn. And every week, over and over, this was happening. So these are smart bears. They just were like, okay, give me my food. I'm just going to sit here and wait for it. Thank you very much. So here's kind of a cute picture that kind of describes the story of what these bears are doing. So here we have a bear coming in. It's probably been attracted by the lure that's hanging in the trees. You can't see that. And it crawls over the barbed wire. So you can see that it's going to step over that. It walks around and it eats all of the remaining corn, but then it still smells that stuff in the trees. So it's curious. It wants to figure out where that's coming from. It goes up, it pulls down the lure and messes with it a little bit to finally get at that smell. And then it leaves. And this is the same trap. And the next day we have a mother and a cub that comes in. So you can see that this did attract a number of different individuals over several days, and we got quite a good representative sample of the individuals found in these areas. And then ultimately, what did we find in terms of the results? We found that in Pittsburgh, after running algorithms and counting the number of individuals from the genetic analysis, there was about 80 individuals, black bears there, and in Milan, there was about 95. And this was actually really interesting because the Milan area had more bears, but it was more of an urban setting. And so it's interesting that it was a higher number of bears that was in this area. And I strongly suspect that because it was a slightly more urban, I say urban, but we're talking about kind of rural, suburban New Hampshire, but it's not an empty forest, I should say. But there was some development, a little bit of town kind of system. And in this area, there are a lot of resources that humans end up bringing to this area. So, they have garbage, we have bird feeders, you have growing things like maybe some apple orchards or some berry bushes or something like that. And so I suspect that area could support more individuals because of that human influence on that system. And this kind of relates to this next graph I'm going to show you, which I think is something else that was interesting. Now, what we're looking at in this graph, each of these dots here are a different bear individual, and this is representing them in space where they were actually found. And the bubbles, the larger the bubbles, indicate clusters of high relatedness and of females. This is a graph that just shows females. So when we look at Pittsburgh up here, we can see that there's kind of 3 clusters of related females, and they're kind of spread apart, right? They're more spread apart. And then in Milan here, you can see lots more overlapping clusters of these related individuals. And that suggests that, one, in this area, the offspring are not moving very far from their parents. So that's one interesting thing. And also, because those clusters are closer together, it means that there must be more resources available to support that many individuals in that area. So I think that also is probably linked to the idea that there's more food resources in those areas. Okay, so now we're going to switch organisms again, and we're going to talk about amphibians and the pros and cons of studying amphibians. So one of the pros is that you can, as you can see, yes, you can hold them. So another check on the can you hold them wildlife biologist checklist. On the top here, you can see a baby metamorph wood frog, and they're very cute when they are first developed after their larva. So that's another check for pros for amphibians. And on the bottom is a picture of a spotted salamander. And I just can't get over how crazy the whole salamander thing is. I mean, it's like a giant slimy lizard thing. I don't know. I love it. It's just like so out of this world, so alien. So another reason to love amphibians, because it's like aliens. And speaking of pros and aliens and out of this world, one of the really cool things that you get to do when you're studying amphibians in the way that I was studying them is you get to put on these full-size waders. So you have your feet, and then the waders come all the way up to your chest, so they're chest waders. And then we were walking around these vernal pools, so these kind of small but deep pools, looking for egg masses, and that picture on the bottom is egg masses. But while you're walking around in these waders, it is uncanny. It feels like you're walking on the moon, because you're kind of like floating up, and the water is all freezing cold around you, and it's smushing you in, and you can't go very fast, and you're like, so it's exactly like moonwalking, or how I imagine moonwalking would be. And that's another pro for amphibians. Now, there are some cons to studying amphibians that's very important to keep in mind. One of the cons is that amphibians like the water, and they lay their eggs in water sources that are also where mosquitoes lay their eggs. And because they like water, they also are most active during rain events. And so when you are studying amphibians, you have the slightly uncomfortable situation of being out in the drenching rain. If it rains, you have to go outside, you have to go and catch them even more than you would have to catch them regularly, because they're more active when it's raining. and you are standing next to the most giant, incessant mosquito source in the entire forest. And so here's a picture on the bottom here. You can see, even though I'm kind of half smiling in this picture, it's the middle of the summer, it's sweltering hot, I'm wearing long pants and long sleeves and a head net. And that must mean that the mosquitoes are so bad that the head net came out. And that's a serious contention right there. So on top of that, when we were studying them, we were studying all of the metamorphs that were coming out of the pond. And we would be catching hundreds of these at a pond. And so we would be out there in the rain, in the sweltering heat with the mosquitoes, measuring hundreds and hundreds of these tiny baby salamanders. That is a con for studying amphibians. And then this picture up here is another kind of advice. If you are studying amphibians and you have to drive around in rainy events in the middle of spring, it's very good to have a trusty vehicle. Four-wheel drive is important. And I can tell you from experience that you may want to purchase a winch for when you're driving around in these muddy, unpaved roads. You will get stuck and you will have to call AAA, and it will take them about 4 hours to come and get you while you're sitting there thinking about how you're missing your sampling event and you have to come out tomorrow and you have to get to the salamanders. And so a winch is a good tool to have to use those trees to pull yourself out. And we did have to do that on more than one occasion. So another point to be thinking about when you're thinking about studying amphibians. So now to the science related to this study. So in this study, I was focusing on looking at spotted salamanders on top and wood frogs on the bottom. And I was interested in looking at how do they move in the landscape and what kind of physical features of that landscape were impacting their movement, either acting as barriers to movement or acting as facilitators of movement. And so we went out, and as you saw before, we got to wear the moon pants and catch the go and look for these egg masses, and we would collect those. And then I did an analysis using genetic information to try to figure out what types of features impact the salamander and frog movement. So here I have some pictures that were most associated with each species. So up here, the salamander had associations with roads, with kind of clear cuts and power lines, clear cuts associated with power lines, and then clear cuts associated with forest management. And then with frogs, they were associated with forests, other ponds, and these forests, these cut forests as well. But there was some interesting interactions that we didn't expect to go on. So first, I'll tell you about what we kind of did expect, and that is we did expect a negative relationship with movement in gene flow and roads. So this means that if there are more roads in the landscape, there's going to be less movement across those roads. So it's going to kind of act as a barrier to movement. And we also found that to be the case with these linear power line structures for the salamanders. And then for wood frogs, these clear cuts down here pictured were also associated with a lack of movement. And that makes a lot of sense because frogs are organisms that need moisture and require this kind of moisture. And if you have clear cuts, you've lost the kind of forest canopy that keeps that moisture in that system. And so they tend to avoid them when they're kind of moving from pond to pond. But what we found that was interesting was positive relationships. We're going to start with the wood frog here, between forests, and that also makes sense because those forests have a canopy layer that keeps things moist or more moist on the ground, especially after rains. And so when you have areas with more forests, they're more likely to move through those forests. And when you have areas with ponds, the frogs are also more likely to move in those areas because they can use those ponds as stepping stones and go from one pond to the next pond and kind of try to figure out their movements based on that pond. But this was what we found that was really interesting. And the story here is that for salamanders, unlike the pattern with frogs, there seemed to be a positive correlation between gene flow, and clear cuts. And this was puzzling because, like the frog, the salamanders should prefer moist habitats. And you would think that when they got to these clear cuts, they would be maybe turn around or be repelled from them. But we found that there was more movement through them. And I suspect what's going on here is that in this case, when a salamander found itself in an environment that was like this forested clear cut, what they did was they were like, I got to get through this, right? They can't turn around. They don't know where they're going behind them. So they just got to go through it, and they just go. And so it actually adds, it is a positive relationship with movement because it's some kind of maybe motivation for them to move through that really rough habitat. Okay, now we're going to switch organisms again, and we're going to talk about the pros and the cons of birds, and more specifically, the pros and the cons of rails. So this is a picture. This is kind of a rare picture, and that is of whether or not you get to hold birds. So one of the pros is sometimes you do get to hold birds, and if you're lucky, you can set out bird nets and hold them. In this case, the rails that I was studying are super elusive and secretive, and it was very hard to capture them, as I'm going to tell you about in a minute. So it's pretty surprising that I did get to hold one of these birds. One of the other pros about studying birds is that you get to join this like wild and wonderful world of this group of people. They're called ornithologists, and they like wake up for the crack of dawn and go out and sample birds and go look for birds that they have on checklists. And they're really excited about that. And it's kind of neat to join in on that excitement. So that is one plus for studying birds. I would not call myself an ornithologist, so I just kind of jumped in that realm and then jumped out again. Now, I want to talk a little bit about some of the challenges to studying organisms too. We kind of think of, if it's out there, you can catch it, right? You can find it. And that is not always the case, as exemplified by my experience with these rails. So we had to figure out, we were going to do some genetic analyses on these, and we had to figure out how are we going to catch them. They live in marshes that are flooded, so they're just giant mud flats with grasses up way past your head. And there hadn't been done a lot of research because they were so hard to catch and so hard to find. So one of the ideas that we had was to use this picture here. It's a clover leaf trap. And the idea was we were going to put this trap in the marsh and then build like fences along to the edge of the trap, the opening of the trap, these big giant kind of silt fences. And then we were going to use acoustic technology to broadcast the calls of the rail, and then hope that because males are territorial, they would come rushing in to defend their territory and go right into the trap. And I should mention that when you're usually trying to trap organisms, it's good to put a food reward, but there's no way that these organisms needed a food reward because the food resources were everywhere in the mud, anywhere they went. So they weren't going to be attracted to this trap by the food reward. So we were like, okay, we're going to get them through their territoriality. So here's kind of a picture of us setting this up. We had to get a weed whacker and just cut marsh grass because we couldn't even see and couldn't even get anything in there because it was so knotty and wild. You can see that we are standing in lots of mud. You would be standing in mud to your knees, and then you would take a step and you would be up to your shoulders in mud. It was not a very clean experience, and kind of weird too, because when you're in that much mud, you don't know what's down there. You can't see anything. Could be anything, any organisms down there trying to bite your toes off. It's a little unnerving. But this completely didn't work. We couldn't get a single rail to come in here at all. So we had to rethink it. This is another idea that we had thought about. It is kind of like a mouse trap, but giant. And we decided to try to use this. So again, here you can see in this picture, we have the acoustic songs being played, trying to get those territory males to come and stand somewhere on these boards. And then the board would be triggered by that, and this net would fly over them and hopefully catch them. And you know what? this again also did not work. These rails were like, what are you, do you think we're stupid? Of course we're not going to be walking up on this board, coming to this other thing. They were like, no, thank you. I'm gonna stay hiding in the grass. So again, it didn't work. Something else that I can give you advice about is if you happen to be carrying one of these giant traps on the back of your truck, make sure you tie it down really well so that it doesn't fly off on the highway and smash into the car behind you. Not the thing that we had experienced quite like that, but very, very close to that. So again, keep that in mind. So it turns out that none of those things worked. So how are we going to catch these rails? Well, we did some research and we found kind of an interesting article about how some people had success catching rails, and we decided to try that out. So what we did was we first researched the highest high tide and of the month, and then we went out at the full moon. So that's going to be where it's even the highest high tide, right? And we went out on these hovercrafts. So in these marsh areas, it slides even higher on the high tide, it floods even higher on the high tides that have a full moon. So we use these hovercrafts, and at midnight or whenever the fullest moon was and the fullest tide, and we zoomed all on top of these marshes using a thermal camera, looking into the marshes to see little blotches of red that would indicate the clapper rails. And we used a net and we would catch them that way, and we got our highest trap success from that. So we caught 27 birds in one night, whereas we had caught 0 birds and using any other technique. And so that turned out to be the thing to do. Now, the tricky part of that is that you can, when you're using the hovercrafts on the marsh, it can push down the vegetation. So you need to make sure that you're avoiding things like the breeding season. You do it earlier than the breeding season, because otherwise you're just going you might go over their nest and devastate that habitat. The marsh grass bounces back pretty quickly though, because it is very resilient. Okay, so now the science behind what we were doing in this project. Here we have a picture of a mated pair. This is a mated pair of 2 different individuals from different species. So on the left, we actually have a king rail. You can tell by this orangish tone. And then on the right, we have a clapper rail. You can see by the slightly more gray tone. And so these are 2 different species that have mated together and presumably produced hybrid offspring. When you look at the king and the clapper rail ranges, here we have the king on the left and the clapper rail on the right, you can see that for the most part, they have very similar overlapping ranges on these coastal marshes of the southeast. And so it can be tricky to distinguish them because they live in similar areas, they eat similar things, they're in similar habitats, but it turns out that they can be distinguished by the level of salinity or salt in those marshes. So clapper rails are more associated with high salinity marshes, and king rails are more associated with low salinity marshes. And one of the questions that we were interested in is, are there places in Virginia where these 2 species are mating? When you go out, you can hear them calling everywhere. And as I said, you cannot see them or find them or catch them, but you can hear them calling, but you cannot distinguish the king rail from the clapper rail via their calls alone. So we were trying to figure out, can we use genetic methods to distinguish these? And is it possible that there's hybridization going on in different areas? So this is the result for that. We had enough samples to do a comparison between this more inland site here and this more salty kind of site, I want to outland site here. And we found out that when we looked at it, that the Pamunkey River site was found to have 4 out of the 36 sampled individuals that were king rails. So 11% of that population appeared to be king rails, as compared to the other site at Mockhorn Island, where only 7% of those individuals were king rails, so 1 out of the 14. So this does support that there is a salinity gradient that is being followed, and that the king rails are found at slightly lower salinity areas. And it also suggests that there is likely hybridization between the clapper and the king rail in that system because they're found in the same exact sites. And I did find further evidence of hybridization using other genetic tests. All right, so this is where I'm going to kind of switch things a little bit. I have thought a little bit about this, and I am thinking back to kind of to my background and how little I knew when I was reading those National Geographics as a 10-year-old about what field work and that kind of field adventure would be as a woman. And so I have some advice for future female field scientists that I like to share. So number one, be a good judge of character. Number two, find a supportive advisor. Number three, don't let yourself be humiliated. And number four, say yes. So I'm going to explain all of these points of advice that I have and kind of some stories that go along with these. So my first story, be a good judge of character. This story is related to when I was studying abroad in Tanzania. I went over there and I did, as an undergraduate, and I did a semester studying wildlife biology in Tanzania, and we saw giraffes, lions, and gazelles, and it was pretty amazing. And at the end of that field study, the way they had worked it out was that every student was to write a proposal to do their own research project anywhere in the country by themselves. So I decided, I'm thinking about this, what would I want to do? And it turns out that my advisor back home in Texas had studied a species that was found in this area called the sengi, or the elephant shrew. And this is a rare mammal that is actually more closely related to elephants than to other shrews. So instead of calling it an elephant shrew, there's been a push to call it a sengi. It looks small in this picture, but it's about the size of a giant rabbit. So it's like this giant furry rabbit with a long elephant nose that's eating insects in these forests. And since my advisor had studied them, I was always kind of interested in them. And I decided to do a research project where I went to see, where can they be found and what kind of habitat do they like to live in. And so I basically set off by myself with a month's worth of supplies, took buses to remote villages, and then from there took mopeds, just hopped on mopeds up mountains to this other remote village, and then got off at that remote village and kind of asked around. And in that process, I was trying to essentially find a forest wildlife officer who was supposed to be living in that area and talk to them about doing a study. And in the process of asking around, and I kind of guess, there's a white woman, a Wazungu in the village, what's going on? And it turned out that I ended up meeting a family that was an expatriate, so an expat from Sweden. It was a man from Sweden who had done a lot of Tanzanian work helping out Tanzanians. And he had a Tanzanian wife, and some children. And they hosted me for a day or two while I was trying to figure out where is this forest officer and how can I get into the jungle by myself. And when they found out what I was doing, they were like, I don't think you should go by yourself. And I was like, okay, I don't know. And so they said, they kind of talked amongst themselves and they said, we want our family friend, this man named Tobias, to come with you and stay in the forest with you. And I was like, okay. And it turns out when I actually talked to Tobias, he used to hunt these sengi when he was a youth. And so he kind of knew what to look for and how to find them. And so I was like, yes, this is great. I would definitely like help with this. And so they sent me off, and I eventually did find the forest officer. And we arranged for me to do this kind of small scale research project where I was going to study these. And I took off with Tobias, me and Tobias, and we walked 4 hours from the village to the forest, the Chome Forest Preserve, which is where we were going to try to look for these. And I remember that day. I didn't know how long it was going to be to the forest. It was but it was still exciting, and I was young and I could hike all day. So but I actually remember, this is funny, and I'll give you this story too. I actually remember that this Swedish couple helped me, or the Swedish man helped me, because it turned out when I was staying with them, I saw this kind of dot moving around in my foot, and I was like, that's weird. I have a dot moving around on the bottom of my foot. And I showed it to them, and they were like, oh, that's a flesh-eating parasite. I was like, oh, that's great. And so they helped me take it out. We had to take a needle, we burned a needle, and then we dug this hole in my foot. It was like an inch deep, and then pulled out this parasite. And they kept saying, like, it was like this long worm, and they kept saying, we need to be careful, because if we split it, it'll stay in your foot and you'll have an antibiotic reaction to it, And so, like, okay, let's make sure we get all of that worm out of my foot. But I did this 4-hour hike on that foot, and it was painful. I remember being very, very painful. Anyway, but I was still excited. I was young, I was adventurous, I wanted to find these fungi, and I was excited. And we didn't actually get to an area outside of the forest to set up our field site until late in that afternoon, maybe like 1 or 2. And so we set up our field site. This is actually a picture from the field site. We had a big tarp because it was going to rain on us, and we had separate tents. And we had, that's where we were going to camp out for the next 2 weeks and see if we could find these. But that day I was like, but I want to go and see if we can see now. So I convinced Tobias that we needed, even though we hadn't ever been, neither of us had ever been to this particular place, that we should go check out this forest. And so we hiked down the trail and into the forest, and we're kind of walking around and looking at stuff. And I think, I'm not sure whose idea it was. It's a little murky at this point, but one of us decided to go bushwhacking into the forest to see if we could look for these giant leaf nests that these sengi build, because that's what we were ultimately going to find. And we did not have any luck seeing giant leaf nests that day, but I did start to notice that it was getting darker and darker. And then I started to notice that there was like this cacophony of noise in insects, just like insect noise, like at that dusk of insects. And I was like, okay, maybe we should be heading back now. But we had started bushwhacking and we were lost. We had no idea where we were. We couldn't get back to that main trail. And he was leading around, and we had this long giant machete, and he was like swiping things, and I was following him. And then I was like, forget this, I'm leading, and I was swiping things and he was following. Eventually we did hours after hours, and getting darker and darker and darker, and thinking like, nobody is going to find me here. This is not the best place to be right now. Whose idea was this? Stressful. It was stressful. We eventually found the trail back, and at this point I was leading and we were kind of going through, and it's like this this bushy trail and lots of things and everywhere, and I like ran headfirst into this really sharp stick, I guess, and I was like, oh, and then like I reached up and like gushing blood everywhere down my face, and like we still hadn't gotten to the campsite. It was like half an hour away, and I'm stressed already, and I'm mad that we're in here. I was like, what is going on? And if you look closely at the picture, you can see that blood, that blood gash that I gave myself. So that was actually not what I wanted to talk about when I'm talking about be a good judge of character. What I really wanted to talk about, in a little more serious note, is that the scenario here became uncomfortable fast. So Tobias was a young male, probably in his early 20s, and I was a young female in college, and he pretty quickly decided that he was going to start making advances and trying to talk about, do I have a boyfriend, and when am I going to get married, and maybe I should marry a Tanzanian person. And about 3 or 4 days in, he had proposed, like honest to goodness proposed marriage. And I didn't know what to do, right? I'm alone in this forest with this guy, 4 hours from the nearest village, and he had the machete. So I remember at night, I would listen to the sounds, like in the middle of the night, whenever I heard like his tent unzip, and I would like jolt awake, and I would be like, and just hold my breath until I heard it go back in, because I didn't know, I didn't know what he was going to do. I didn't know what was going to happen. I didn't know where that machete was going to be. It was really scary. And I guess ultimately, when I say be a good judge of character, I trusted those people who did offer him as a guide. And I was right to trust him because he was a gentleman the whole time, but it didn't mean that I wasn't scared. And it didn't mean that I wasn't vulnerable. And so I do recommend females who are thinking about adventures in the field to be a good judge of character. All right, on the second note, the second advice that I have is find a supportive advisor. So there can be very wonderful male and female graduate advisors. And you should, if you're interested in field work, you should take the time to go find the ones that are going to be the most supportive for you. So I want to give you a story here about the situation where, so in my master's program, when I was doing the bear research, so I was going out and I had 2 different field sites and I had 4 different field techs. And the 4 field techs, they were all male, and I had 2 males at each site. And they would kind of do stuff, and I would be going back and forth. I would spend like a week there, and then I'd come back and do a week there, and I was collecting all the samples, and I had all the data sheets, and I was organized, I was on top of everything. They were just going out and kind of collecting their hairs and having fun. They were doing a lot of having fun. And one day, the Fish and Game liaison, who had been working closely with us, who was the bear project leader, he came out with us, and he was me and 2 of those field techs. And he spent the entire, he must have come out with us for a couple of hours, and he was like chatting to, basically he just was chatting to the guys the whole time. And he was asking them things like, oh, how many samples do you have here? How many samples did you collect last week? How many samples are at the other site? And these field techs had no idea, because they weren't in charge of the project. They weren't collecting those samples. They were just kind of there, you know, doing things. They weren't keeping track of any of this stuff. And I remember sitting in the back, kind of hearing this and getting uncomfortable and being frustrated, because I wasn't being acknowledged as the leader of this project when I was leader. It was very much this male rapport happening with these other male technicians, and I was basically ignored. And it was, as I said, it was frustrating. And later that day, I didn't say anything at the time, but later that day, it started kind of bubbling up. You know, I've been taught to be a strong, independent woman. I need to say something about this. I need to let this field liaison, this Fish and Game liaison, know that I'm in charge. And I appreciated him talking to me about some of the stuff, like it's a direct communication rather than talking to these male field techs. So I ended up writing an email, probably in the heat of a strong female, proud female woman warrior, and saying some of these things along those lines. And we ended up having a phone call, and it got a little bit ugly. And basically the story is, for the rest of the time, I ruined that relationship. So I was considered by him, we had like a cordial relationship before, but then after that, he wouldn't talk to me at all. It was very much working through the advisor. It was a very kind of cold, standoffish relationship at that point. And I had 2 advisors on this project, 1 female and 1 male, and the male advisor they must have talked to each other about this, whatever. And I kind of became labeled as a difficult person to work with. And I realized that's tricky. And you know, and this, I think in hindsight, maybe I should have just swallowed it and not allowed myself to say something, even though I thought in order to be a strong feminist, I should have said something. But it did teach me something, that in the future I would kind of bite my tongue or make sure that I wasn't making other people uncomfortable, because obviously that would come to haunt me in retrospect. So that male advisor I lost a relationship with, and that Fish and Game person I lost a relationship with, but I learned some things. And I also, you know, it reinforced that wildlife biology is definitely a male dominated world. And so they're quite cordial, they're quite inviting, but oftentimes the men tend to socialize more with each other, and they congregate and have kind of a more jocular relationship with each other. So something to keep in mind as well when you're coming into this wildlife profession. Think about that kind of, it's a man's world. It very much is in this scenario. All right, don't let yourself be humiliated. All right, so this one is kind of also related to, you know, being a female and being a female field scientist. So first of all, there are certain situations that only females will recognize. So this is why I say find a female advisor if you can. They're going to understand what that male culture is like. They're also going to understand what it is like to be worried about your safety. And they're also going to understand what it's like to be camping with a period. And let me tell you some advice. If you're a female field scientist, A, the Diva Cup is a great thing to have, or B, the OB tampons that don't have any trash. So definitely think about those. Very important life lesson. If nothing else, take that home with you. But also, there are other things that come up as being a female in the field sciences. And this relates to my project where I was working on the marshes. And on the marshes, when I was doing this, I was a postdoc, and it was, we were scheduled to have a month in the field working to find these clapper rails because we couldn't find them. Remember, we talked about that already. And it was me and a female graduate student. And then we had these two kind of, again, a liaison from the Department of Fish and Game, and they were friends, and they were helping us out. And they would drive the boats out around the marshes as we were looking for things. Now, I was a postdoc at this point, and so I did this field work with a 6-month-old baby. And we actually couldn't find any site to stay in when we were doing this project. And so the only thing we could find to stay was essentially, it was a garage, like an actual like commercial garage where it had like the doors that go up, and it had like things that would hang from the ceiling in order to move cars up and down. And it was sweltering, no air conditioning, just these giant doors. And I brought my 6-month-old with me because I was breastfeeding. So we were living in these conditions, and it was hot and sticky, but it was an adventure, and I always was up for an adventure. So here we go. And then every day I would go out on the field, on this boat. And here's a picture of me on the boat. And you can see the kind of jocular, funny stuff that was happening with the guys on board. And that was fine. They were fun. They were respectful. But we would get into a rhythm, and we would be going out and setting up these traps and putting things down. And then every 4 hours, my watch would buzz, and I had to stop. I had to tell everyone that I needed to stop, and I had to pump. And it was, in retrospect, when I think about it, it was humiliating. I would sit there, I'd have to say, we need to stop everything right now. They would turn around on the boat. We were in the middle of nowhere. We couldn't go anywhere. I had no privacy. I would just turn around so that my back was to them. And then I didn't have any electricity, so I had to hand pump for like 40 minutes in order to, get the resources needed. And then these guys are joking behind me. I don't know what they were joking about, but I'm almost certain that at some points it was joking about the situation. And then I had to like button up and be like, okay, here we go. Let's go again. And I just, at the time, all I could think about was, don't think about this too much. Just keep doing it. You need to do this. Here you go. This is what you're going to do. Don't be humiliated by it. And at the time, I was really good. I was in a good mindset. I was like, whatever, I need to do this. It needs to happen. But later, the humiliation was just so strong. Just not having any privacy, being in this middle with these two older men who were laughing and joshing around. It was humiliating. So don't let that stuff get to you. If you're a female in the field, just keep going, know what your goal is, and enjoy that. And then finally, my last piece of advice is say yes. Say yes to all the opportunities that you do get and will get. You go and step outside of your comfort zone. Go on that swinging rope bridge if you're afraid of heights, because you're going to learn so much about yourself and the world around you. So if I didn't say yes, I wouldn't have gone to Tanzania and lived in a Masai boma for a couple nights, where it was probably the most miserable experience in my life, in a sweltering hot boma with the fire going, choking smoke, sleeping on a giant wooden bed with bed bugs crawling over everywhere, hundreds of bed bugs crawling in every single orifice of your body, with 4 kids on one side and then the mother on the other side. If I hadn't said yes, I wouldn't have been able to experience that. If I hadn't said yes, I wouldn't have gone to Indonesia on a wildlife backpacking trip to see the wild orangutans of Indonesia, and I wouldn't have gotten to see this particular picture of this mother and this baby coming down from the trees when they heard us approaching, reaching out and inspecting what we were doing there. We could almost touch them. They're just like giant 8 feet tall furry beasts. I mean, it was phenomenal, and I wouldn't have been able to do that if I hadn't said yes. If I hadn't said yes, I wouldn't have gone to Japan and taught English for 2 years after my undergraduate degree. I knew I wanted to be on National Geographic and I knew I wanted to go back into science, but I also knew I wanted an adventure first. So even if you think you do want to get somewhere in the end, it's okay to take a little bit of detour and explore the world first. If I hadn't said yes, I wouldn't have climbed to the top of Africa's tallest peak, Mount Kilimanjaro. That's me on the left-hand side, even though you can't quite tell, at a time when there was still glaciers there, and there probably won't be glaciers there in the near future. And if I hadn't said yes, I wouldn't have licked a slug in British Columbia on a dare and found out that the sticky parts of the slug slime goes into your cells on your tongue and holds on and makes your entire tongue numb for 3 hours. So say yes, you'll learn a few new things. So good luck and have fun. And with that, I'm done.
All right, we're going to go ahead and open the floor for questions. Yeah, so we'll just open the floor. If you've got a question, just unmute yourself, and we'll take that as you are trying to ask a question.
Think of something to say.
Would you actually like to say it?
Mm-hmm.
Okay, go ahead.
Um.
It has to be about the talk, though. Okay.
I really like to talk.
Oh, thank you.
Can I make a comment as well? I would like to say that your lessons were good lessons, and I think reflect a lot of thinking on your part too. But as somebody just looking in and hearing them now, I actually think that you're a good role model for speaking up and being a strong feminist. And I wish that you wouldn't feel humiliated and so forth now, because I think those are really brave and strong things to do in the time.
Thanks.
Well, I'm going to jump in because so you have touched on so many different animals, and I just kind of was curious what's next in terms of research. Are you going to touch on reptiles? Are you going to get into that realm, or are you going to stray away from that?
Well, I have never been a huge reptile person, but you know what? That doesn't mean I'm not up for it. So oftentimes what I do is I work collaboratively with people who have questions that they need answering. And so sometimes people come to me. Recently, Hellbender Project has come to me and asked me to help them look at stream restoration efforts and see how Hellbenders are doing. I've also had the Fish and Game Department here come to me and ask me to look into nutria, so invasive mammal species, and see whether or not we can track their spread and how to eliminate them. So those, and then I've been also working with crayfish recently, and then I also have been working with pollinators. So that's one of the cool things about what I do is that because I use genetic techniques in all these different scenarios, I don't need to be an expert on the organisms. I mean, I learn a lot about them as I go, and I enjoy that. But I use the same techniques, and because of that, I have a lot of freedom to do those same techniques with a lot of different organisms. And it's all about the adventure.
Thank you. That's awesome.
Any nests while you were doing the field walk?
For the rails, did you see any nests?
Oh, I think one time we did. I don't know if you saw that picture, but I was holding up a little round ball, and that was a nest that we found. We only found one nest, and it had an egg in it, and I took the egg because at that point we hadn't been able to catch anything, and we were desperate. So I took the egg, and we used that as one of our samples. But no, it's really hard to find them. They're like in the grass and all tangled together, and when you step in that mud, you just sink and you get stuck, and it's like a big sticky grass thing, and it's a hard place to work.
Since no one else is asking questions, I'm going to just jump in again. Have you has there been continuation of any of these projects, and maybe with yourself, maybe with others? And do you know, like for instance, is there more information known about the hybridization of the rails, or has hunting started in northern New Hampshire, and how have the populations responded?
Good question. I have not continued to work with these. Certainly the Bear Project, I may have burned that bridge there with the Bear Project by talking about, how I should be acknowledged as the leader of the project. So I don't know about how that's going, but that makes me want to look into it. So I should do that. Rails are, as you saw, are hard to catch. So there's some studies that have been going on with king rails, because there is one population that can be kind of approached, but I haven't seen much about clapper rails because they are so hard to study. In fact, another part of our project was seeing if you could distinguish the vocalizations between the king and clapper rails, because that would help identify them. And it turned out you can't. So that makes it even more tricky. You really do have to kind of catch them in hand or see them with your eyes to figure out what species they are. And it's really hard to work with those. Yeah.
So you've talked about the sampling, and I thought it was funny the way you described your first few efforts, and then your final successful effort seemed pretty elaborate. Did you come up with those sampling processes, or were those things that do researchers just kind of come up with stuff, or is it written out for people?
That's a good question. With the rails particularly, it was just a lot of people brainstorming, like, what in the world are we going to do? How are we going to get this? We knew we couldn't use bait. That's the original thing. You always think, like, what kind of food bait can you use? We knew that wasn't going to work. So then it was like, well, someone said, there was a lot of talking to people. So-and-so said that one time they caught something using this, and then so-and-so said, try that, and then so-and-so. So that's kind of what we did. We kept desperately trying until we could figure out something that worked. And in the end, I was surprised. That hovercraft scenario, it worked really well. I was surprised, but also it took a lot of resources. We had to find the hovercraft. We had to get this group of people out in the middle of the night, and it had to be so there's only one there's only one high water every month with a full moon, and then you had to not be raining because you couldn't be out there in the rain doing all this stuff. So a lot of things had to align to get that just right. That is one pro about working with birds, though. When it rains, you don't have to study them. So if you're interested in birds and you don't want to go out in the rain, there you go. You don't have to go out in the rain with them because they hide out too.
All right. Do we have any? We're getting close to our time. Do we have any more questions? I'm going to open it up just for one more question. All right, well, I'm gonna go ahead and share my screen real quick. Dr. Koster, thank you so much. This has been awesome. I did want to thank Lainey, Dr. Rollins. They really are instrumental in all the stuff that we do as far as putting this together. So we are appreciative to all those who showed up. We will actually have this on our website here next week. So we'll have the video up. So if you want to tell your friends about it or anything like that, you're more than welcome to. We invite you all to come next week as well. We are having our final installment. It's Close Encounters, Field Adventures of a Wildlife Biologist with Dr. Carlos Rojas. He's a professor of biosystems engineering at the University of Costa Rica. Thank you all again for joining us. Find some adventures. Tell your friends to come and join us. And thank you again, Dr. Koster, one more time. Thank you. Bye, everyone.
Thank you so much, Dr. Koster.